Hello everyone, welcome to my channel. Hope you guys are doing well. In today's video, I'm going to discuss about Coagulase test. In the microbiology syllabus, there is a topic named food poisoning, and in questions, most of the time, it comes as a Staphylococcal food poisoning. Now, to detect Staphylococcus aureus for Staphylococcal food poisoning, the laboratory test done is Coagulase test. So this comes as a short note. So without getting any delay, let's get started. Now let's talk about what this test is all about, right? So Coagulase test is a very important test. It basically detects Staphylococcus aureus from other Staphylococcus species. This test differentiates Staphylococcus into two groups. One is Coagulase-positive Staphylococci and the second is Coagulase-negative Staphylococci. Now, coagulase is basically a protective barrier for those Staphylococcus. And if these coagulase is present in their cell wall, basically what it does, it increases the virulence of that genus and it provides a resistance, against the human immune system. Coagulase directly interacts with the fibrinogen present on host cell surface. This Coagulase test is basically done in two ways. One is tube Coagulase test, two is slide Coagulase test. What are the various specimens that collected in Staphylococcus infections? These are pus, sputum, blood and urine. Now, pus is collected when there is suppurative lesions and in case of osteomyelitis. Sputum is generally collected in case of respiratory infections, blood in case of bacteremia, and urine in case of urinary tract infection. Let's talk about the first process, that is tube Coagulase test. Now, in tube Coagulase test, as the name suggests, it is done in a test tube. It is carried out to detect free coagulase. In this test, 0.1 milliliter of an overnight broth culture is mixed with 0.5 milliliter of 1:10 dilution of human or rabbit plasma. The plasma is then collected in vials containing anticoagulants, that is heparin, oxalate or EDTA. The plasma broth culture mixture is incubated in a water bath at 37 degrees centigrade for 3 to 6 hours. Now, in case of a positive test, plasma is coagulated and does not flow. Now, here you can see if coagulase is present in staphylococcal cell wall, when the test is done, there will be a clot formation, right? And then if the clot is formed, we can say that the Staphylococcus genus is Coagulase positive. Now, here one thing is to remember that citrated plasma is not used in this case, because if the specimen is contaminated with gram-negative bacilli, that may utilize citrate and produce a false positive reaction. Let's talk about the second process of doing the Coagulase test, that is the slide Coagulase test. Now, as the name suggests, the test is done in a slide. It detects bound coagulase or clumping factor. The test is performed by mixing a dense suspension of Staphylococci with a loop full of undiluted rabbit plasma on a slide. Now, in a positive test, clumping takes place around 10 seconds. Now, here you can see in the slide, in case of Coagulase-positive Staphylococci, there will be a clot formation. So, it is a Coagulase-positive Staphylococci. Now, let's talk about some Coagulase-positive organisms. The most common is Staphylococcus aureus. Other than that, there are Staphylococcus intermedius, Delphini, Hicus, Lutre, etc. Now, see some Coagulase negative organisms. The most common in this is Staphylococcus epidermidis. Other than that, there are Staphylococcus werneri, hominis, capri, etc. Now, see some advantages of this test. It determines the presence of coagulase in the microorganisms. It also determines the production of coagulase by different microorganisms. And it differentiates Staphylococcus aureus from other Staphylococcus species, which is very important in detecting the virulence and also to correctly diagnosing the condition. Let's see some disadvantages or limitations of this test. The first is methicillin resistant Staphylococcus aureus can be deficient in bound coagulase, which results in a negative slide test but a positive tube test. Now, in case of Staphylococcus intermedius and Staphylococcus hicus, may be positive in the tube test, as these species are generally found in only dogs and pigs respectively, but these are as infectious as Staphylococcus aureus when they infect humans. Staphylococcus lugdunensis and Staphylococcus silpheri produce slight coagulase, but the reaction is more efficient if the human plasma is used rather than rabbit plasma. Coagulase testing cannot be performed from growth on mannitol salt tablet. That's all for the topic. Thank you so much for listening to the video. And if you like the video, then please like, share and subscribe to the channel. And you can also join the Telegram account. Link will be given in the description below. Thank you so much. See you in the next video.